Bismillahirrahmanirrahim. My dear student of second year class, in the previous lecture we have described only the introduction of the GIT tract, their functional aspect. Now we discuss further the structural organization on the basis of histology of the GIT. Histology of GIT almost comprised of 4 layers in all the viscera and the lining, but these 4 layers are variable according to the region and structural topography. So these 4 layers all the 4 layers comprised of mucosa, submucosa, muscularis propria, serosa or adventitia. Now the mucosa consists of epithelium, lamina propria, muscularis mucosa. Now these all 3 layers constitute the mucosa. So the mucosa is present in all the viscera or the tubular structure, but the epithelium is almost the same, but the lamina propria is variable, having at some places glands and at some places it will be structural position or structure containing connective tissue. Muscularis mucosae is also a constant feature starting from the hollow tube, that is the esophagus, up to the almost anal canal. The epithelium is the starting from the stomach up to the large intestine, that is the ascending, transverse, descending sigmoid. The epithelium is simple columnar epithelium. The muscularis mucosae is a smooth muscle layer which encircle the mucosa and also separate from it from the submucosa. Submucosa consists of the loose connective tissue containing all the elements, that is the fiber, cell and the ground substance. The fibers are the collagen and elastic fibers, very little amount of reticular fibers. All the cells of the connective tissue are mostly fibroblast. Then the macrophages, lymphocytes, plasma cells are also visible. The characteristic feature in this is the mesenal submucosal plexus. So these are the autonomic nerve plexus, especially the parasympathetic coming from the myenteric or orbital plexus. These plexuses are also very important as far as concerning the secretions of the various glands which are pouring their products within the lumen of the GIT. These plexuses maintain the secretory activity of the GIT. The muscularis propria or the muscle layer, this is again a constant feature, and these muscles are smooth in type, encircling the tubular GIT, circularly arranged and longitudinally arranged, and in between the circular and longitudinal arranged, these are the plexuses of the orbex, which are also called as myenteric plexus, very important because they are concerned for the motility of the GIT, and they are also having very important neurotransmitter mechanism through which it also regulates the motility according to the situational activity of the GIT, according to the human personality, the mood, and also various hormonal influence is on the GIT motility. the serotonin, acetylcholine, substance P, all the neurotransmitters are concerned, and also motivate or also changes the motility of the GIT through this mind direct plexus. Serosa or adventitia, these are the very much constant features in the whole GIT. Serosa is the peritoneal lining, little bit connective tissue lined by the simple squamous epithelium, whereas adventitia, where there is no peritoneal cavity, it is the loose connective tissue surrounding outermost layer of the GIT. This is the diagrammatic presentation. You see, this is the tubular organ starting from the esophagus up to the anal canal, the epithelial lining inside, and then it is the lamina propria. This is the muscularis mucosae. Now, this one is the submucosa. Now, look at the glands. These glands in the submucosa also move. These glands are the mucosal gland, which are in the lamina propria. The epithelial lining is simple columnar, though in the submucosa the glands are characteristically present in the duodenum. Then is the muscle layer, inner circularly arranged, and outer is the longitudinal. In between lies the blood vessels and the mind direct plexus. The submucosal plexus and mesenal plexus also located in the submucosa. Now, these are the nerve and artery moving inside toward the muscle layer and submucosa. The outermost is the adventitia or the peritoneal. This is the serosa, because the mesenteric branches of the mesenteric vessels and nerve plexus from the mesenteric plexus move inside from this mesentery into the muscle layer and then into the submucosa as well. So these are the four layers: the mucosa, submucosa, the muscle layer, and outer serosa. So you will classically remember all these four layers, but always remember that this structural organization is in the tubular organ. Somewhere it will be different, but you will have invariable. It will be present in the form of a mucosa or muscle layer and serosa, but it will be variable in structure topographic. Now we start with the oral cavity. The oral cavity starts from the lips up to the palatopharyngeal arches, from where posteriorly the oropharynx will start. Now in this picture you see these are the lips, and then these are the palatopharyngeal arches, palatoglossal arches. So from this palatopharyngeal arches up to the lips, this whole is the oral cavity. The oral cavity is comprised of the vestibule and then the oropharyngeal isthmus, which is lined by the palatoglossal and palatopharyngeal arches, and this is the oropharyngeal isthmus, posterior part of the tongue, the uvula. This is the soft palate. Over here will be the hard palate. So these are the teeth, socket and teeth, the incisor, then is the lateral incisor, canine, premolar and molar, and this is the palatine tonsil, tongue and the vestibule. These are the frenulum of the tongue, then the papillae of the sublingual and submandibular gland, salivary gland, and then the gingiva. So all these structures will comprise boundaries forming the oral cavity. Now we come to the histology of the lips. You all know the lips is the skin tag and enfolded in the oral cavity, from where there will be a definite landmark from the skin and the mucosa or the mucous membrane of the oral cavity. This line is called as vermilion border. In this diagram you see the transition between the skin and the mucous membrane of the oral cavity is the vermilion border. Same in the case over here, the skin and then the mucous membrane you see in this diagram. The histology of lips could be comprised of the outer skin, then the lamina propria is comprised of terminal papillae, sebaceous gland, hair follicles, inner oral mucosa, then skeletal muscle, scattered salivary gland. So this is all are the features will be present in the lips. After the skin, at the pelvian border, there is a transition from the mucous membrane and then the skin. You all know that the skin is lining with the certified squamous keratinoid epithelium with the hair follicles. Sebaceous glands are also present. So all these features are present in the skin. But at the pelvian border, there will be acute transition between the skin epithelium to the certified squamous non-keratinized. Certified squamous, that is typical oral cavity epithelium. The hair follicle will be not present, but the feature of the same certified schema, but it is the non-keratinized. Then the mucosal, the lamina propria also contain the salivary skeletal glands, and the innermost layer is the skeletal muscle, always in orbicularis oris. This is not a smooth muscle, already told to you in the previous discussion. Over here, the outermost covering is skeletal muscle, not the smooth, because these are the voluntary movements of the lips. So skeletal muscle will be present in this histology lip. Look at the picture. So this is the certified squamous non-keratinized epithelium. So it is the transition between the lip, orbicularis oris, to the muscle layer, the skeletal muscle. Then the labial mucosa, that is one of the mucosa of the labial mucosa, certified squamous non-keratinized. Scattered salivary gland, you see in the groups, in the bunches of labial salivary gland. This is the hair follicle, sebaceous gland. This is skin. So this is squamous keratinized epithelium. You see hair follicle, then sebaceous glands are also visible. So this one is the lip skin, and this is the lip skin, and then this is the certified squamous oral mucosa. So this is the typical picture. You will have to describe it as a outer skin element, all the feature, then inner mucosa, which is this mucous membrane of the oral cavity, then the skeletal muscle and scattered salivary gland. यहाँ पे हम बेटर है कि हम ग्लैंड्स की बात कर लें. जो ग्लैंड का लफ्ज़ आया तो ये आपको all together GIT में we will have to discuss the glands. So it is the gland is the epithelial modification. Epithelial modification present within the epithelial tissue moves into the mucosa, submucosa. They are meant for synthesize a substance such as a hormone for release into the bloodstream. When it is bloodstream, then it is called endocrine, or into the cavity inside the body. It is outside surface, it is called as exocrine gland. या वो surface से उसको pour करेगा या फिर cavity के अंदर, it is called as exocrine by well-defined duct system. It is called as exocrine. Endocrines are those glands which have a well-defined system, that the secretions in the form of a hormone that are released directly into the bloodstream, no duct system. No endocrine gland के example जब आपके हैं तो आपको very well known that the hypothalamus, pituitary gland, the parathyroid gland, then the thyroid gland, then the thymus, kidneys, the suprarenal gland, pancreas, and the islets of Langerhans, the best example of endocrine gland. Then the ovaries and testes, the interstitial cells or ladies, and the theca interna and externa, they are concerned for the release of progesterone, estrogen. So all these hormones pour their secretion directly into the blood vessel, not through the duct system. So these are called as endocrine glands. Now this is the examples of the exocrine gland. You look at the picture, that is the modification of epithelial tissue, moving either in the form of a tube, in the form of acini within the mucosa or submucosa. Unicellular, for example, glands are the goblet cell is the best example. They are present in variable amount in the GIT, starting from the small intestine and up to the very prominently present in the large intestine up to the anal canal. Respiratory passage, it is the characteristically present in the respiratory passage, that is the trachea and bronchi up to the respiratory bronchiole. So goblet cells are usually present within this, because they are meant for secretions of the mucoprotein, and this mucoprotein is concerned for the lubrication, and then secondly it expectorate out and also trap the various microorganisms. This picture is the simple tubular gland. Simple tubular gland, they invaginate the epithelial tissue within the mucosa. Those simple straight glands, it is characteristic feature of the stomach and colon. Simple branch tubular abundance, single tube divided into two, and then the secretory unit become tubular, double tubular. Then it is called as simple branch tubular gland. Lower portion of the stomach में आपको नजर आएगा. Simple coil tubular gland, stomach and small intestine में in the form of coil formation. It takes turn. Although it is a epithelial modification, but it takes turn. Simple acinar, sebaceous gland in the skin. Simple acinar means that it has a well-defined tube, but it has a acini, rounded ball-like structure lining by the secretory cell. Pour the secretion first in the lumen of the acini, and then this will move toward the duct to the surface. So this is the best example of sebaceous gland, simple acinar gland. Simple branch acinar, this is again example of a sebaceous gland of the skin. Multiple branches of the acini are present, draining into single duct. First pour the secretion lumen, then into ducts, then into the single main duct toward the exterior. Compound tubular acinar glands, mucous glands of the duodenum, look at the picture. These are multiple ducts are present having the tubular gland, but there are no acini, but these elongated tubules, secretory tubules, so they will multiple join to single duct, then single to the major larger duct. This is an example of a duodenum. Compound tubuloacinar glands, example of mammary gland, look at the picture. Tubules are also present, also the acini. Acini, tubule, smaller ducts, then larger ducts, ultimately in the excretory duct. The next example is the acinar gland. Then in the compound tubular acinar, so it is like the pancreas, both acini, then the duct system and major ducts, they are also example of a pancreas. So this is the according to tubule and according to acini, हम इनको divide करते हैं, and these are at some places it is present. So this is the examples for you for the exocrine gland, because they pour their secretion directly into the either exterior or into the lumen via duct system. This is again a mode of secretion. हम इसको divide कर सकते हैं glands को into three types. Mode of secretion. Secretion कैसे करते हैं? Endocrine example है sweat gland की. In this, the acinar cell, they first produce their secretion within the cell, intracellular, then with the help of vesicle exocytose within the lumen of the acini, then move into the duct system. This is called as endocrine. कि हम पहले उसको secretion जो है वो सेल अपने अंदर रखेगा, with the help of vesicle at the time of need ये secrete करेंगे exocytosis within the lumen of the acini, then move toward the exterior. This example is the endocrine. Apocrine gland में अगर mammary gland के example दें, in this case some part of the cytoplasm along with the secretory vesicle No, in the halocrine, in the apocrine glands, you see some part of the apocrine gland, some part of the products of this cell surface, cytoplasm surface, also pour into the lumen of the gland, lumen of the acini, then into secretion. So partial necrosis of the apical cytoplasm. Necrosis means that is sloughing of the cytoplasm into the lumen of the alveoli also present. Then is the halocrine gland as an example. In this halocrine, you see the sebaceous gland is the best example, from where the whole cell along with secretion is poured into the lumen of the acini. So all secretions first stored in the form of a this secretion along with the whole cells cytoplasm, also called, this is called as the holocrine, and this is the sebaceous gland example. So these are the types of the mode of secretion according to the mode of secretion, types of the gland according to the mode of secretion. So this is all about the today lecture. Next we'll move on to the further oral cavity structures. Thank you very much.